My research work is primarily in the chemistry of carbohydrates, or more loosely referred to as the chemistry of sugars. Carbohydrates, also interchangeably referred to as saccharides, are a major group of biomolecules that have many applications in living systems, whether it's sweetening up your coffee in the morning or acting as essential cell communication molecules for the maintenance of homeostasis in multicellular organisms. There are a variety of common carbohydrates, such as glucose, fructose, and galactose. Carbohydrates are almost exclusively formed from carbon, hydrogen, and oxygen. However, different atoms can be synthetically attached. Now, nature uses simple monosaccharide carbohydrates, such as the ones that I've already listed, to make more complex carbohydrates, which thereby allows for more complex functions to be carried out. My research primarily focuses on working to synthesize two different carbohydrates, one being a monosaccharide, the other being a disaccharide. Both of these molecules are glycolipids, which means they have a polar sugar head and a nonpolar fatty acid tail. Now, in nature, the golden rule is structure determines function. The structure of these carbohydrates allows them to exhibit nanoparticle self-assembly behavior, which effectively allows them to bypass the nonpolar lipid membrane of the cellular plasma membranes in the tissues in which they target. These carbohydrates have been isolated originally in Streptococcus pneumoniae, a deadly pathogenic bacteria that mammalian organisms have struggled to survive with throughout evolutionary time. Invariant natural killer T cells are a defense mechanism that have evolved to deal with threats such as Streptococcus pneumoniae. Invariant natural killer T cells produce a nonspecific systemic immune response that is designed to target threats in the body such as bacterial infection. Now, motivation to synthesize the two carbohydrates that I've spoken about is through studies which show their ability to increase the activity of invariant natural killer T cells in cell culture. What this means is that these carbohydrates can have applications in human pharmaceutical medications because they can be used to initiate a very powerful immune response against viral infected or tumor cells. Now, before these applications can be further studied, these molecules have to be synthesized. One of the major problems that we have to deal with in the chemistry of carbohydrates is the reactivity of the hydroxyl groups found on the backbones of the monosaccharides. These hydroxyl groups can undergo many different reactions, and in the reaction schemes that must be carried out, a variety of protection and deprotection reactions must be carried out so that the proper regioselectivity of the reagents used can be ensured. Now, despite the difficulty, the synthesis of the monosaccharide is almost complete. To make the more complex disaccharide will require a very much larger reaction scheme involving likely 21 steps. The motivation to make these compounds will likely lead to not giving up to synthesize them.